Hello. In this session, we're going to talk about cell death. Specifically, we're going to talk about the different types, the morphologic types of necrosis, and how it differs from the different types of cell death. We're also going to talk about the second type of cell death, which is apoptosis. And then we're essentially going to compare the two and describe instances where necrosis happens and when apoptosis happens. Now, this slide is essentially a review of everything that we've talked about so far, and we will be focusing on this part here. What happens to the cell when it reaches the point of no return and the injury becomes irreversible? Over time, the cell is going to die due to the persistent or severe injury, and cell death occurs as a result. Now, the most common type of cell death that occurs secondary to cell damage is called necrosis. Necrosis is a type of cell death, and as what I've explained earlier, this is a type of necrosis where basically the cell is being eaten from the inside out. The enzymes released by the lysosomes and the proteasomes digest the contents of the cell. It denatures the proteins, and the plasma membranes get digested out, causing leakage and lysis of the cells. This injury is irreversible. Once it reached that point of no return, um, there's really nothing. There's really not much you can do. And because of the leakage of the substances out of the cell into the surrounding environment, necrosis is usually dirty in the sense that there is associated inflammation. The leakage of substances outside the soil will actually incite WBCs to go into the area in a process known as chemotaxis. Necrosis does not just happen to a single cell. It happens to multiple cells in the area. Of course, because this is a type of cell death, there is always a loss of function of the involved organ. And as mentioned, the leakage of substances causes inflammation and the migration of WBCs into that specific area. The release of enzymes when necrotic cells die is actually the basis of how we measure um, certain laboratory analytes or certain chemicals in the blood to measure or to diagnose certain diseases. Examples include troponin I and CKMB, which are markers that we use, which are chemical markers that we use to diagnose myocardial infarction or heart attack. Okay? Now, there are two types of lysis involved. It's either the lysis of the dying cell due to the release of enzymes from the lysosomes, and this is known as autolysis, or it could be due to the lysosomes of the surrounding dying cells, or the lysosomes that are secreted by WBCs that migrate into the area, and this is known as heterolysis. Regardless, these two types of lysis, autolysis and heterolysis, work hand in hand to destroy the cells. And as mentioned, this is the type of cell death caused by disintegration of cells. Okay. Now, if you leave tissue lying around outside the body, over time, the cells are gonna die on their own, and this will manifest microscopically as necrosis or autolysis as well. Now, there are many types of necrosis, and the most important that we need to know are these five. Okay, but the most important actually are the first two, coagulative and liquefactive, since these are the two most common that we will be seeing. Okay, but we have five types: coagulative, liquefactive, caseous, fat necrosis, and fibrinoid necrosis. Now, coagulative necrosis occurs during ischemia. Okay, when does coagulative necrosis occur? Coagulative necrosis occurs as a result of ischemia in all parts of the body except the brain. Okay, let me repeat that. Coagulative necrosis occurs in ischemia in all parts of the body except for the brain. Okay, now if you have an area of coagulative necrosis, you call that an infarct. Okay, so if there's an area in the kidney that undergoes coagulative necrosis, usually it's the tubules that undergo that first. Um, you call this a renal infarct. If it's in the heart, you call that a myocardial infarct. If it's in the spleen, you call it the splenic infarct, and so on and so forth. Now, how does it look like? Essentially, areas of coagulative necrosis tend to be lighter in appearance, and they are well demarcated, okay, and they tend to be firm. Okay. Now, how do they look like? And if you take a look at this picture here, so this is an area of the kidney. Okay, so you have this wedge-shaped infarct. It's yellowish in color. It's lighter in color. It's well demarcated, and if you feel it, it's actually quite firm compared to the rest of the kidney parenchyma. If you look at it under the microscope, there is a pale area as well, a pale pink area. And if you take a closer look, you will find that you will only have outlines of the cells. You cannot see nuclei. Okay, they are called ghost cells or tombstone cells. Okay, so it loses cellular detail. You just see the faint pink outlines of the cells. Okay, so that's coagulative necrosis. Okay, so this is what it looks like under the microscope. So here you have the glomeruli and then the tubules here on the left. Okay, now if you look at the high power magnification, you will see that you have these very pink um, tubules, and you have actually a glomeruli here, but you cannot see nuclei anymore. So they are called ghost cells, okay? And this is characteristic of an infarction, okay? Another example of necrosis, here you see liver parenchyma. You have hepatocytes arranged in plates or trabeculae, okay? And here in the middle, you can see an area where you still see the faint outlines of hepatocyte trabeculae, but you don't see nuclei anymore, and it tends to be a bit more pink than the surrounding cells. So this is coagulative necrosis of the liver. And as mentioned, you see this in ischemia, except the brain. Okay. A second type of necrosis is liquefactive necrosis. Now, whereas in coagulative necrosis, there is leakage of proteolytic enzymes and digestion of the contents of the cell, the plasma membrane has not been digested just yet. That's why you will have the outlines in coagulative necrosis. In liquefactive necrosis, everything is digested, including the plasma membrane. So you do not see ghost cells with the faint outlines anymore. Instead, you will have this amorphous pink material, okay? And no outlines anymore, okay? And this is again due to the release of digestive enzymes and its accompanying inflammatory process, okay? So where do we see liquefactive necrosis? We see this in two major scenarios. We see this is in ischemia of the brain, okay? So this is the only exception when ischemia is involved. And number two, you see this in bacterial infections. Okay, you see this in bacterial infections. Now, in these instances, the cells get digested. Not only do the lysosomal membranes get destroyed, but also the entire cell, including the plasma membranes. You will see amorphous or a formless pink material, mga abo-abo, with associated inflammatory infiltrates like neutrophils. Okay? And over time, these necrotic cells are gobbled up by the phagocytes by the body. Okay, so this is an area of liquefactive necrosis. Okay, see here, it's wet, it's mushy. Okay, unlike yung firmer appearance of coagulative necrosis. And if you look at it under the microscope, it's going to appear this way. So amorphous, walang shape at all. You don't see any cells, pink, dusty material. And you tend to have a lot of inflammatory cells, particularly neutrophils. The third type of necrosis is a type of necrosis that is seen in infections that cannot be cleared right away by the body or by the immune system. You typically see this in infections that are not easily cleared. Okay, and the most common example would be TB. Okay, the WBCs in the body cannot actually kill TB bacteria because of the chemical composition of the cell wall of the mycobacterium of the mycobacteria. Now, caseous necrosis is a special type of necrosis that is seen in long-standing infections, including but not limited to TB. And it is called caseous necrosis because of how it looks like grossly. Caseous, cheesy. So it looks like cheese. Okay? Yellow, curdy. Okay? Ano siyang cheese nga maragidugmok. Okay? Now, what happens actually in long-standing infections, for example, in TB, in fungal infections, is that they do not get killed by the neutrophils. So what happens is that the body is going to shift since the body cannot be killed. The body cannot kill it through acute inflammation through neutrophils. The body shifts to chronic inflammation. What it does is it tries to enclose it with more powerful WBCs. Okay. Now, chronic inflammatory cells called histiocytes, called macrophages, will envelope these organisms, and they will try to kill it. They will try to envelope it. As part of the process, the macrophages will try to fuse with one another to form giant cells. Okay, and they will all secrete their hydrolytic enzymes to try to kill the organism. Okay, now this necrotic material, lined by a rim of macrophages, lymphocytes, and other chronic inflammatory cells, is known as a granuloma. Okay, it's known as a granuloma. So the caseous material containing the organism plus the inflammatory cells, particularly macrophages and giant cells, all of these constitute a granuloma. Okay, so this is how it looks like grossly. So you have this yellow cheesy material. This is actually a section of the lung. Okay. Now, another type of necrosis is known as fat necrosis, and this is a special type of necrosis that involves adipocytes or fat cells. You usually see this in acute pancreatitis, where leakage of enzymes from the pancreas, particularly lipase, is released into the abdominal cavity and digests the fat. Okay, so lipase splits the fat into glycerol and free fatty acids, and then the free fatty acids will combine with calcium to form calcium soaps or white chalky material. Now, acute pancreatitis is scary because it releases digestive enzymes, it digests everything, and it causes massive hemorrhage. That's why acute pancreatitis is very dangerous. Now, how does it look like grossly? Okay, so you have this white chalky deposits. So this is areas of fat necrosis in the mesentery. So this is actually due to the release of lipase into the abdominal cavity. The fat is split to form salts, to form soaps with calcium. Okay, so these are actually calcium soaps or calcium deposits. Microscopically, this is the normal adipocyte. So you have fat globules, and then the nuclei are pushed towards the periphery. Okay. Now, how does it look like under the microscope if it undergoes fat necrosis? So you have this on the right. Very pink granular material. You only see the outlines, but you don't see the detail anymore. Okay. Now, the fifth type of necrosis is known as fibrinoid necrosis, and you see this typically in blood vessels. Okay, there is deposition of a protein called fibrin in the arterial walls, and this is usually associated with two groups of diseases. Number one, persistent severe hypertension, known as malignant hypertension. And number two, you also see this in autoimmune disease, or the deposition of autoimmune complexes in the walls of the arteries. So as mentioned, they usually occur in the arteries. They typically deposit. It's how does it look like? It appears as pink material, bright pink material in the walls of the blood vessels. Okay, and this is usually accompanied by neutrophils. Now, the gangrenous necrosis is not actually a histologic type of necrosis. It's more of a morphologic description. Okay, so gangrenous necrosis is essentially necrosis applied to an extremity. Okay, necrosis applied to an extremity. Now, if the necrosis is due to ischemia, if the gangrene or necrosis to an extremity is due to ischemia, then that is known as dry gangrene or mummification. Okay, and how does it look like? Essentially, it's dry, mummified siya. Okay, that's dry gangrene. Now, another type of gangrene is when you have coagulative necrosis. When you have liquefactive necrosis to an extremity that initially had dry gangrene. So of course, in dry gangrene, diba, there's ischemia, the tissue is going to die. Okay, so coagulative necrosis. But over time, if you have dead tissue and it doesn't get taken away, it doesn't get amputated, the dead tissue is going to undergo inflammation, it's going to undergo lysis, diba? Bacteria is going to eat it. Okay, so from a coagulative form necrosis, there is going to be a superimposed liquefactive necrosis. And once that happens, the process becomes known as a wet gangrene. Okay? Now, the second type of cell death is known as apoptosis. And in contrast to necrosis, which occurs due to damage to the cells, apoptosis is said to be programmed cell death. It is genetically programmed form of cellular suicide. Okay? And it results from two pathways, is called intrinsic and extrinsic pathways. And essentially, it's a series of chemical reactions that will tell the cell that it needs to self-destruct. However, self-destruction is not due to lysis. The body likes everything clean and orderly, so instead of lysing these cells, the apoptosis tends to package cells into smaller, denser structures to be gobbled up by the WBCs. Okay? As mentioned, apoptosis tends to be physiologic or pathologic. Now, when do we see physiologic apoptosis? So there are organs during the growth of an organism in the embryonic stage that will only serve a certain purpose up to a certain age. And after a while, the body, during its development, will no longer make use of it. So during these instances, these vestiges or mga unneeded organs will undergo apoptosis. Okay? Another example is during menstruation cycle or in the breast in response to the cyclical nature of estrogen and progesterone. So depending on whether or not a particular hormone is present or withdrawn, then this will facilitate a cycle of growth and then apoptosis. For example, in the endometrial lining of the uterus in the normal cycle of the menstruation. Okay. Apoptosis may also be seen in proliferating or actively dividing cells. Cells that have undergone their that have performed their functions also die. And sometimes the body may produce self-reactive lymphocytes. And once the body detects it, it's going to trigger the lymphocytes to undergo apoptosis so that it will not produce autoantibodies. Okay. Now, when do we see apoptosis in pathologic conditions? We see this in case of injury. Okay, like for example, chemical injuries like radiation. Apoptosis may also occur in certain infections, particularly viral infections. However, instead of causing the cells to undergo lysis, these viral agents will integrate themselves into the genome of the organism and trigger apoptotic pathways instead. This is particularly true in hepatitis viruses, okay, and um, in certain in certain infections like hepatitis and infections of the skin. Okay, atrophy in certain organs, especially in organs that can be blocked, where you have secretions that can be blocked over time. If there is prolonged blockage of the ducts, the organ will undergo apoptosis. This is particularly true for the pancreas. Um, the thymus also undergoes atrophy. This is normal, but if you accelerate the process by giving corticosteroids to a child, the thymus is going to undergo a more rapid progression of the atrophy, and this will lead to apoptosis as well. Now, how does apoptosis happen? So, when the death pathway genes are activated, this triggers apoptosis, and essentially think of it as the cell shrinking itself. So, chromatin condenses to become more dense, and it undergoes fragmentation called karyorrhexis. So, initially there is condensation or pycnosis, and then there is fragmentation known as karyorrhexis. Okay. However, the cell's plasma membrane remains intact, unlike necrosis. And then, since you have the production, you have the end result here are more dense, smaller cells. What's going to happen is that these cells are going to get phagocytosed by macrophages. Okay, so here you have these apoptotic cells here. So these are actually Councilman bodies, small arrows, itong pink dense material. Okay, so those are Councilman bodies seen in hepatitis, and this is actually this is a reflection of apoptosis within the liver cells. These bright pink material are actually apoptotic keratinocytes seen in certain dermatoses. Okay, like mga lichen planus. Okay. So these are important enzymes and important genes that are triggered in apoptosis. Okay, and this is a schematic diagram differentiating necrosis where there is lysis and apoptosis where you have more condensed cells at the end of the process. Please also note that in apoptosis, you don't usually have associated inflammation. Okay, so that ends our lecture on cell death, and this wraps up everything for chapter 2. Thank you very much for listening, and good luck in your exams.